Okay, well, we're going to continue this next lecture by getting more into some of the basic physical and biological structures that you find on salt marshes, specifically in New England. And these are the commonmost species that you find. So going from right to left, at the top you have Spartina alterniflora, that's also known as cordgrass. You have Spartina patens, which is also known as salt meadow hay. Disticus baccata, which is spikegrass. And then at the bottom you have Juncus gerardii, which is called black rush. It's a very beautiful plant. Limnonium carolinum, which is sea lavender. And then Salicornia europea, which is the slender glasswort. These are just some common plants that you can find in these habitats. Now, one of probably the most striking organisms that you see in most salt marshes throughout New England is this very large Phragmites australis. This is called the giant reed. And there is an invasive Phragmites as well that makes this kind of look like a miniature version. So it's unique that a Phragmites has come in and has invaded and has basically, taken over and outcompeted a lot of the more local species. But as far as some of the animals go, there's the Littorina littorea, which is the common periwinkles that you all saw at Lighthouse State Park. There's Zostera marina, which is an eelgrass, which is quite common, just subtitle of salt marshes. There's Ilionassa obsoleta, which is the eastern mud snail, which should not be confused with the Littorina littorea. And then there's the organism that we talked about at Lighthouse, the Crepidula fornicata, where it settles as a male, and then if no females come along, it goes through sex change and becomes a female, and then the males settle on the back of the shell. And you also have other soft-shelled clams like Maya aranearia, and there's a few other quahog species, etc., that can be found in New England salt marsh habitats. So the other quahog is the Mercenaria mercenaria. Then you have the eastern oyster, Crassistrea virginica. You have the Ucker species of fiddler crab with that very large claw. You have Limulus polyphemus, which is the horseshoe crab, which is only found basically right around now during high tides and full moons. So if you went out to the beach, anytime through May through early July, and it's high tide and a full moon, you're going to see hundreds, if not thousands, of horseshoe crabs on the coast mating. I urge you to do that. It's actually a pretty amazing thing to go see. And just walk along the rack line of that water level, and you're going to see a lot of horseshoe crabs. The upper right-hand column is another type of mussel, a mollusk called Gukenzia demissa, and it's a ribbed mussel, and it lives within the salt marsh. But it does put down byssal threads. You can see byssal threads coming off the mussel there and attaching to the very soft substrate. So with all these organisms that are out there, salt marshes provide a great many free services for us, basically. They avoid flooding or runoff. So by having plants that trap nutrients, etc. It acts as a carbon sink because of all those plants photosynthesizing, so that's a direct loss of atmospheric CO2. It acts, as I mentioned earlier, for estuaries. It also acts as a nursery ground for many commercial and recreational fish and shellfish. There's microbial processing of organic and inorganic waste throughout these habitats. And you have erosion control by binding sediments. So the fact that you have these plants there acting as sediment stabilizers, you essentially avoid the erosion of sediments out, into a deeper area of the water. So these are some of the free services that we get from salt marshes. Now, these characteristic animals that live in the salt marsh, well, it all depends on whether or not it's a high tide or a low tide. So what you see is the lower area of the marsh at high tide at the top part of this graph. And you can see all the different species that are present, some of the ones that we mentioned. But then when the low tide happens and the water gets really, really low, you see an entirely different group of organisms coming in. And, the rib mussels will always be there. The amphipods will always be there. But you got a lot of insects. You have a lot of mud algae. You have a lot of blue crabs and fiddler crabs. But then you see some bird species and raccoons. I know it seems really odd to have just a picture of a raccoon there as being a very common low marsh organism, but it is. When that tide goes out, that opens up a lot of those ribbed mussels for those raccoons to eat. And I think along the coast of Connecticut, the marshes have single-handedly maintained the raccoon population. So the different types of species you find is entirely up to the time that you go out to these sites and whether it's a high tide or a low tide. Because if it's low tide, you're going to get very different species. Hence, it makes sense to visit both during a high tide and a low tide in order to do species counts or looking for any abundance in species. Now, when we look at the zonation, now we talked about zonation patterns in rocky intertidal, and those are those striking lines that Stevenson and Stevenson first described in 1929. You can see them at all different geographic levels. And you can also see zonation in a New England salt marsh. So if we go from the low marsh, where it meets the water, to the high marsh, you have the ribbed mussels, the Gukensia demissa. You have cordgrass, which is Spartina alterniflora. And you have some fiddler crabs, burrows. And then as you hit the high marsh, the cordgrass gives way to Spartina patens, which is the salt hay zone. You can get some slender glasswort, the silicornia. You can also get some salt meadow hay, which is the Spartina patens, that continues throughout that zone. And then that mixes with some of the black rush zone, the Juncus gerardi. And that continues all the way through. You get the marsh elder zone. And the reason they call it marsh elder is that those are the plants that have probably been around the longest. They're the elder plants. Because the water doesn't really make it up there that much, but they're highly resistant to the habitat that they're in. And those include plants like the Iva frutescens. So you have this very characteristic zonation pattern of a New England marsh. From Spartina alterniflora to Spartina patens to Juncus gerardi to the Iva frutescens, and then if you just look at the bottom, you see a repeat of that, except for the Gukensia and the fiddler crab burrows. Now there's many other species that might be present in here, but this is just the species that make up that very striking zonation pattern. Now this differs than a southern marsh. Okay, so marshes down south, if you remember, marshes are much larger as you hit South Carolina, Georgia, North Carolina, and Florida, right? So it makes sense to kind of compare these a little bit. So we just saw zonation in a New England marsh, and this is zonation in a southern marsh. So what you have is, a same kind of zone. You have the Spartina alterniflora close to the coast, just like you would in New England. But then there's this salt meadow, which is Spartina patens, and that's fine. But then you have this area called the salt pan. Then it picks up again with the salt meadow zone, and then you have the southern marsh equivalent of the Juncus gerardi, which is the Juncus romerianus. Now, the striking difference between the southern salt marsh and a northern salt marsh is that salt pan. So what happens there is that as the tide comes in, water hits that salt pan area, and probably just beyond it to the right. And then when the tide goes out, the salt gets stuck in that habitat. And you'll see this at the West Haven beach that we go when we have our last field day together as a class. It's a very striking, very dry dry area. And if you can imagine, putting salt on something and just letting it sit there again and again and again and again. So the species that might be present in that salt pan have to be incredibly tolerant of a very high saline environment. So this salt pan is a very characteristic aspect associated with the southern marshes. And then if we go all the way to the west coast, we see different species, Spartina foliosa, Scriptus robustus, different Salicornia species, Jumea species, Disticus spicata, and Limnonium californium. And this is the characteristic aspect of a west coast salt marsh, is the fact that it has Limnonium californicum as one of the elder marshes or elder marsh plants that are present. There's usually a high diversity of species in California, the Pacific coast, in relation to the east coast of the United States. But what you don't find here, as you would in the southern marshes, is that characteristic salt pan. Now, these patterns of zonation in these salt marshes around our country are determined by both biological and physical factors. And those physical factors include the same things that affect, rocky intertidal zonation patterns. The flooding or the tidal range, the salinity, any kind of ice scour that might take place, any wrack or dead floating material, fire, as well as sedimentation. The biological factors include competition, either within or between species, and predation and habitat modification. So sometimes predators can come in here, wipe out entire sets of plants, which would change the habitat for that, particular salt marsh. So if we look at a typical Long Island salt marsh, and we go from the water, there's some fish in the water, there's some blue crabs, other types of crabs as well. Then you have the Littoraria errata, which mitigate out of the water with the tide. The Lateraria are the snails that are on the Spartina alterniflora that's present there. So you have the Spartina that can be submerged, but the tops of it stick out of the water, and the snails, in order to not get eaten by the crabs or the fish, actually move up to the top of the blades in order to avoid predation. So then you have some amphipods that are present in those habitats. You can see the ribbed mussels that are there. You might get small juvenile shrimp and fishes that might be in some of the more tide pool-like habitats of the salt marsh. And then in the upper marsh, you have other organisms like Melampus, you have some Gukensias, and then you have the Spartina patens that help set up, the middle marsh area for Long Island Sound. So you have all these critters interacting with one another in these habitats. Now, organisms that are in a salt marsh have to adapt because they are part of estuaries, and we know estuaries to be highly variable. So there are morphological adaptations. Usually estuarine species are smaller than their open ocean relatives. The vascular plants that are present have arenchyma, which is a tissue that supplies roots with oxygen. And that Spartina alterniflora actually has sweat glands. And think about that Spartina alterniflora, it's right on the water line. So when the high tide comes in, it's totally affected by, salt water. And so it makes sense that that plant has glands in it that help push salt out of it so it doesn't get too much salt and dry out. A lot of the plants that live in those habitats, especially those salt pan habitats, actually develop what's called a succulent strategy. And this succulence is a mechanism to maintain a high water concentration. If you just Google succulent plants out there, you'll see what they look like. And, they're very characteristic. They're a little bit wider. They hold a lot more water because any water they might get might be the only water that they have for some period of time. Now, there are physiological adaptations that take place. They have to osmoregulate. And that brings up, two types of organisms and how they do this. There are osmoconformers that are mostly marine, and they can't control their internal salt content. So they can't really tolerate any kind of changes. These are more of the stenohaline species that you saw on that graph from the previous lecture. And they include things like starfish, sea urchins, sea anemones. And then you have osmoregulators that can control their salt content. And these are mostly estuarine species like polychaete worms, some mollusks, some crustaceans. So you have, both of these species, osmose conformers and osmoregulators, live in these habitats, but they totally differ on how they can control or not control their internal salt content. So if you look at some of these adaptations, there are behavioral adaptations as well. The Eucopugnax crab that's pictured there in the upper right-hand area is a type of crab that uses that large one claw, and they use that claw to actually burrow into the soft sediments. And they live in those soft sediments. So if you go to a salt marsh during low tide, you can look along the coast and see these holes in there, and you think, wow, what's that? Is that just the water? Is it like, areas where organisms live? And most of those organisms that live there are the crabs. Now, there are other behavioral adaptations that take place specifically in crabs. And if you look at blue crabs, per se, they have a special migratory pattern. So you have an adult crab, okay, you have a migrating female crab with eggs that go from upriver to the estuary. So these things would be coming down the Connecticut River, or Housatonic, or Quinnipiac. They would migrate as females with eggs into the estuary. They would release their eggs, release the larvae in the estuary, but close to the open ocean. So they would go all the way out fairly far into the sound to release their larvae. Once the larvae is released, it can go into the open ocean or more saline waters where they start to develop. So these larvae need to develop in higher salinities. Then they go through their stages of development from the zoea stage to the larvae stage to the megalopa stage, where they then become young crabs that migrate back into the estuary. It is the adult crabs that you catch along the, Long Island Sound, and in the rivers of Long Island Sound. And it's because they have this behavioral adaptation where they need the salinity for their young in order to survive and grow and reproduce. But then they do a majority of their living in the river and the estuary. Now, what affects these kinds of systems? Well, estuaries and salt marshes are usually close to where human populations live. And human populations, build houses, they put in septic systems, they affect the water table, they plant plants, they have farms, they use fertilizers, etc. So what this figure demonstrates is that there is a constant flow of nitrogen from local habitats, where we all live, into local bodies of water like the Long Island Sound. And the graph there shows the nitrate in the water column and the houses per hectare. And what that demonstrates is that as the number of houses increases, the amount of nitrates in the water column increases as well. So you have this process of constant nitrification taking place in Long Island Sound and in local waters. You can imagine what that does to phytoplankton, right? It can cause blooms. It doesn't necessarily hurt plants because a lot of plants like to use the nitrogen that's present there, but still it comes at a cost. Probably one of the best examples of this is in Greenwich Bay, Rhode Island. Greenwich Bay is a semi-enclosed body of water. It's a bay, and it used to be literally made up of a few towns. Warwick, Rhode Island, and then across the bay from there, Greenwich, Rhode Island. Now, Greenwich, Rhode Island is very rich. Warwick, Rhode Island was very poor. But Warwick, Rhode Island was the place where all the rich people from Providence, Rhode Island used to go to vacation to get close to the water. So it wasn't that far away, and in a short period of time, you could be right on a beautiful bay. But And after World War II, a lot of the soldiers came back, and they all built, as part of welcoming them home, these small 1, 2, 3 bedroom homes right on the water that had septic systems in there. And those septic systems, so let's say they were built in the late 1940s, started to fail in the late maybe 1980s, early 90s. And so you had a lot of nitrogen going into Greenwich Bay. And all that nitrogen produced a massive phytoplankton bloom that caused the quahog population, all the fish, crabs, lobsters, and snails in Greenwich Bay to just wash up on shore dead. There was not much left alive in those habitats because the nitrogen increased the amount of phytoplankton. They utilized all the oxygen in the habitat, and then they died. And then they went through the decomposition process, which actually used up more oxygen, smothering almost everything that was alive on the substrates, and hence killing them. So it's been a really long time of recovery. A lot of those smaller home septic systems were replaced, usually with government grants at great expense to the families that own those homes. And at one point they wanted to tie in with the Providence sewer system, but that was voted down because they thought they would overwhelm that system as well. So is this possible to happen in Long Island Sound? Of course it is. I mean, just look up and down the coast. There's not many areas along the coast that aren't entirely filled with homes, probably using septic systems that could easily affect and cause local phytoplankton blooms. Now, in these marsh areas, there's a lot of different food webs that take place and a lot of different trophic levels. So you have protozoans, nematodes, anaerobic bacteria, and those things get all used up by different macroalgae. They work their way through the soils. Those can get used by different bottom feeders, whether they typically eat just plankton or whether they eat other fish like top carnivores. So you have zooplankton being fed by them, but you have zooplankton feeding on the smaller plankton, etc. It's a very well-developed food web that includes partly anaerobic, which means these organisms don't necessarily need a whole lot of oxygen, to the aerobic relatives that live in the water. So we talked about the different types of marshes that you can have as far as East Coast, Southern, and West Coast. But there's also other marshes depending on how old the marsh is. And on the left you see a young marsh, which is colonized by colonization of intertidal sediment by cordgrass, which you'd expect. And then you can see the higher marsh is just a little bit of area for a high marsh. Then you have a middle-aged marsh where the marsh plants they baffle out and they increase sedimentation and they help build the marsh elevation. So in that second graph you can see that the high marsh area is a little bigger and the low marsh area is a little smaller. And then you have the mature marsh where there's so much sedimentation taking place there that the low marsh is just this tiny little area that kind of has some drainage, and the high marsh gets much larger. Now, the reason this was studied was because salt marshes were so poorly managed and so poorly taken care of that we had to actually watch them, disappear and then see them slowly make their way back to some kind of middle-aged marsh habitat. What you have throughout the coast of Long Island right now is a mix of young, middle, and mature marshes. You want to go for a mature marsh because that has the most land associated with it, that has the most free services associated with it that we all benefit from. Now, how do we impact the marshes? Well, we dredge them, we remove the sediments, we fill them in. First field day, we were at Lighthouse Point looking to the east, and you saw how much was filled in to put those condos there. We pollute them in different ways, just through household waste. and through oil, gasoline, boat waste. And these things all lead to a decrease in biodiversity. But there are other biological factors that affect marshes probably much more than we do. One of the biggest factors or biological factors that affect marshes are geese, and the second would be simple snails. Now, this is just a graph of the increase in the geese population from 1950 to 2001. And you can see it's almost doubled or tripled or beyond that in some cases. Geese, you see them all the time in open fields. Right now there's a bunch of hatchlings on this campus walking around, so you can see them walking around with their parents. And, a few years back, that plane crashed into the Hudson River, and everybody was saved on the plane because of the very fast-thinking captain that was there. He knew exactly how to land, and it was just a really good example of being a good pilot. But what caused that plane to crash was the geese that flew into the engines. And I thought for sure after that happened there was going to be an open day on geese and goose hunting, but there wasn't. The goose population is only getting larger and larger and larger and having more and more of a biological effect. So I want you to look closely at this picture. What this picture demonstrates is exactly what it says: a geese exclusion cage. So this is an area of a salt marsh that was caged so that no geese could get in there to eat any of that. Now look around that, and you can see in that picture that there's not one other area that looks remotely like it. So if you came across this, you would think, wow, why did somebody plant all these plants here? But it's not that. Those plants would have been present in all those habitats if not for the geese population that visited there and totally wrecked that habitat. So geese have a way of going into salt marsh habitats and just destroying them. Now, snails work the other way as well. So the snails, they're kind of a consumer control of vegetation in salt marshes from Georgia through South Carolina, or South Carolina, Georgia. You see this in New England as well. If snails aren't kept in check by the predators, the snails will exploit this food source. Now, the food source is that fish, crabs, and turtles can, keep them in check by eating them. So we have another figure here which represents direct and indirect aspects of species interactions. So if you follow it from the top down, the turtles and the different crabs have a negative effect on the snails, but the snails have a negative effect on the cordgrass that's present. Hence, the turtle and the crabs has a positive effect, indirect effect, on the cordgrass. So when you have a habitat and you go out to see whether it's healthy or not, you want to find a good representation of all these different species that are present, because you want to make sure that things are kept in check. If the turtles were removed or all the crabs were removed from fishing purposes, the snails would eat through the cordgrass and you would have no more marsh left. It would start to slowly sink into the water, and it would take years and years and years for that to recover. So you have direct effects and indirect effects, again, demonstrated in a simple food chain. So snails are great at avoiding predation, and I mentioned this earlier. When the tide is in, the crabs can get close to the snails, but the snails only have to climb the branches, and chances are they're okay. But what's unique in New England is we have this new invasive crab called Hemigrapsis sanguineus, and that actually can climb up the blades as well. Obviously, if they're too heavy, they can't climb high at all, and they'll just get knocked over and probably not be successful at feeding on the snails. But they have created a very different life strategy for the snails. They just try to climb as high as they can before they start tipping the blades over. Now, these predation factors help set up these community structures. We've already talked about this figure. You've already seen it again. But that predation is really what keeps the organisms present in their particular habitats. It helps them develop what their niche is going to be. If we can go all the way back to the second slide of the last lecture. Now, if we look at specific New Haven area salt marshes, okay. So around 1638, John Davenport and Theophilus Eaton arrived and established the colony of New Haven. The early colonists relied on harvesting that salt hay for their livestock. So that Spartina patens was harvested widely along rivers and along the coast just to feed their livestock with. So they exploited this, and they continuously fertilized the habitats by building farms. In the 1800s, the first upriver dam was constructed on the West River, and between 1766 and the 1860s, all kinds of mills sprung up along the rivers. The Quinnipiac, the Thames, the Connecticut, all these different mill towns kind of started popping up. So we look at this view of City Point in New Haven. Now, City Point is the area where the sound school is, okay? If you drew a straight line from C all the way into the back of that picture, that's where kind of 95 is right now. And you can see some of the, D is New Haven Harbor. You can see some of the aspects associated with that there on the right. But if you look at F and I, right, those were salt marsh areas that were, look at all the homes that were there and present. A lot of those homes are still very present there. But it's very different today if you look at the coast of New Haven. So you have West Haven there. There's Oyster Point there. The harbor of New Haven is just beyond that sandy point there. So from that letter E all the way in. And you can see where Savin Rock is. You can see the small rivers coming down. And so this is, essentially the salt marsh areas along the I-95 corridor. So wherever you see a small river meandering in, look at the extent of the salt marsh around it. It's kind of shaded in the dark. So that's a fairly large area of salt marsh that extended up into current neighborhoods. And then you can see where all the City Point is, and you can see where 95 South is, and essentially a modern map of what the area looks like. But if you look at that West Haven habitat now, there's that long red road, right? That long road there, which I don't even know what it's called. I think it's called like just Ocean Drive or Sea Drive or something like that. Beach Drive probably. And Standy Point is still present there. But see that blue line that says Oldfield Center? Well, that blue line was the line that went all the way up into all the different neighborhoods in the 1850s or 60s. So over time what they did was they capped the area of the marshes and they just filled it in, and pushed it all to that little tiny area that's present right there, which is a huge change in the waterfront of West Haven and New Haven. So New Haven just continued to fill marshes until around the 1870s. And at this point, over 60% of all the highways, the trains, everything, those were all built on filled-in salt marshes. The area near IKEA, all the way up through the greens at Yale, those are all filled in. So you have this long history of just total marsh destruction along the New Haven, highway system. And all those mills that were, functioning, for hundreds of years, they're no longer operational, but a lot of the toxic metals and poisons that they help produce and keep their machineries going are still in these sediments and in these habitats.